Hi everyone, today we're going over Kidney 11.3 in the IB syllabus, and this is going to mostly cover the structure, function of kidney, and two processes, ultrafiltration and osmoregulation. This is not a complete summary of what the chapter is, but it covers the most challenging and basic concepts that they will test. Let's begin with the structure. So you have two kidneys, and they're about the size of your fists. They sit right kind of below, posteriorly, but below the 12th pair of ribs. You can actually palpitate for them. They weigh about 120 to 200 grams, and they have these cool structures on top of them, which looks like pyramids, but these are the adrenal glands. We're not going to be talking too much about them today, but they're really responsible for steroid hormones, adrenaline, and noradrenaline. But they can also be involved in your skin, muscle, and bones, and of course, mood. So right over here, you have a bunch of these renal pyramids, okay? And over here, you have minor calyces, and then you have major calyces, which is right below them. Then you have a renal pelvis, which is where actually all the urine will funnel into. And then, of course, this goes down into a ureter, which will enter the bladder. Notice that there is two blood supplies happening. This is a renal artery. The actual diameter of the artery is a lot smaller than that of this renal vein here. So you can imagine the blood pressure entering the kidney is a lot higher since the lumen is smaller. If we were to take a little cross section, like I said over here, then we can zoom in and see the structure of the nephron. So this would be one nephron right here. Okay, and they have kind of two parts to them. Half the nephron is in the cortex, so this upper part over here, okay, and half of the nephron dips down into the medulla, okay? This would be the loop of Henle right here, as well as the collecting duct. And of course, you have the arteries and veins running across this over here. Here's the nephron. It looks scary, it's not. So let's begin over here. You have a capsule, the glomerulus capsule, and this is basically what extracts the filtrate from the incoming blood, right? You have these arteries coming in and out, and these capillaries have a bunch of blood, and it's going to take only a select bit of that filtrate, right? So then it's going to travel down the proximal convoluted tubule. This is where 60% of water reabsorption occurs. And then it's going to travel into loop of Henle, descending, and then all the way to the ascending loop. And then again, absorb more water into the body, become more concentrated into the distal convoluted tubule, okay? And then we finally go into the collecting duct, which actually, like the name suggests, collects all of the filtrate and tries for one last time to get rid of as much water as possible through aquaporin channels. Okay? So now let's zoom up on a cell in the tubule, and let's say in the distal convoluted tubule. So that would be over here, right here. That this is going to be the basolateral side. Okay, and this is going to be the apical side. Basolateral basically means that it's facing the lumen. Okay, so all of our filtrate is running through this side, and the apical side is the other side of that. So it may be that we have a capillary over here or another cell that we want to transfer the solute to. So with our running filtrate, we have a bunch of sodium that we actually want to reabsorb into the body. So it's going to go into one channel like so, right? And actually sodium travels with a buddy, and that buddy is chloride. So he's going to go from the basolateral side to the apical side, and in exchange, we're going to be pumping potassium from the apical side into the basolateral side, right? So we actually want to excrete potassium, but retain sodium. Ultrafiltration is the first step with the kidneys, and we have a glomerulus, which is this network of capillaries, and we have a capsule surrounding the capillaries known as the Bowman's capsule, which is this here. Okay? The glomerulus, it's a really difficult word to say, has an afferent arteriole, which goes in, okay, and an efferent arteriole, which takes the blood out. Supporting cells in this capsule is actually mesangial cells. They help support. You have then these really cool structures called podocytes, which hold the capillaries in place, and they actually interlock fingers or toes, I guess. Podocytes means feet, like so. Now, when they do this, they make fenestrations for molecules to pass through. However, the fenestrations are not large enough for, let's say, proteins, polypeptides, red blood cells, right, lymphocytes, things that you actually really do want to keep in the blood and not excrete, to pass through. But things like glucose or salts, of course, water, potassium, urea, these can all flow through those windows, and they're actually all collected then in this Bowman space, this big funnel, okay? And they will then travel down the proximal tubule. Remember that the glomerular filtration rate, which is basically the rate at which this whole ultrafiltration is happening, right? The velocity of the blood going through the glomerulus is above 55 in a healthy individual, between 15 and 55 in someone with kidney disease, okay, so chronic kidney disease, and with kidney failure, it's below 15. Yes? So remember these. Let's talk about osmoregulation. So as the name suggests, it's just regulation or control of osmolarity. Okay, and this is all happening in the loop of Henle. So for some reference, this is the end of the proximal convoluted tubule. And on this side, this would be just beginning the distal convoluted tubule. Okay, and now we're in the medullary space. Okay, but what is osmolarity? Osmolarity really just measures the volume of particles per kilogram of solvent. So for reference, usually the proximal convoluted tubule and also the blood has an osmolarity of 300 milliosmoles per liter. Okay, so that's pretty standard if you don't have hypernatremia or hyponatremia. So now let's look at these terms. What does hypertonic, and isotonic mean? Hypertonic just means that there is a decreased osmolarity, meaning lower solute concentration and higher water concentration. Hypertonic is the exact opposite. So you're going to have increased osmolarity with increased solutes and decreased water. And isotonic is where the water is equal to the solute concentration or the salt concentration. The loop of Henle is made of three main components: the descending limb, the hairpin loop, which is the bottom, and the ascending limb. The descending limb is important because it's very thin and it's only permeable to water. So that means through these aquaporin 1 channels, water is able to leave the tubule into nearby capillaries, okay, and be reabsorbed by the body. The ascending limb is a little bit different. They have channels, selective channels, sodium, potassium, chloride channels, which are only permeable to solutes, not to water, okay? So as water travels along the tubule, solutes are going to be pushed out into the medullary interstitium here. Now, because of the hyperosmolarity of the interstitium, right, it's hypertonic in comparison to the descending limb because of the high concentration of solutes. And this actually means that more water is extracted from the descending tubule fluid, okay? And this helps to maintain its hyperosmolarity. This mechanism is known as the countercurrent mechanism, okay? Whereby solutes are being pumped out from the ascending limb, okay? It is changing the osmolarity, which pulls water outside of the descending limb and maintains this reabsorption of water into the body and highly concentrates then the urine. So the filtrate starts over here, right, descending limb. When we first began, like I said, the proximal convoluted tubule, it's about 300 milliosmoles per liter. Correct? And as we go down, we're extracting more and more water, so our filtrate is becoming more concentrated, okay? By the time we hit the hairpin, or the bottom of the loop of Henle, we've hit 1,200 milliosmoles per kilogram, okay? When we go back up, because we're secreting more water than solutes, by the time we actually reach the top of the ascending limb, then we're about 120 milliosmoles per kilogram. Large variables that determine blood pressure and blood volume is the water concentration in the body and also the salt concentration, okay? Salt and water increase blood volume and thereby increase blood pressure. And this, of course, wants to be maintained at a homeostasis within the body. If the body senses that there is a deficiency in sodium, so this would mostly be in the distal convoluted tubule, but also in the juxtaglomerular apparatus, okay, which we didn't cover today, then that will be taken care of through the secretion of hormones like renin, aldosterone, and angiotensin. Okay, and you'll later learn that this is the RAS system. So that's really important for increasing the absorption of solutes in the body. But let's say, for example, that we actually don't need more solutes, we just need more water volume. So when we have a deficiency of water, what happens? Well, a message is sent to the hypothalamus, and in the hypothalamus, the posterior pituitary gland starts to secrete a hormone called ADH, or antidiuretic hormone. Antidiuretic hormone goes to the collecting duct and starts to increase the expression of aquaporins. Okay, and like we said before, aquaporins allow for reabsorption of water. So even when the last stages of the kidney, as much water as we can is going to be reabsorbed into the body. And that's why someone who say they're dehydrated, they will not only pee less volume of water, but their water is more concentrated in dark yellow or orange, for example, because it has a higher volume of solutes. So ADH is a really important hormone to regulate the volume of water within the body, which acts on the expression of aquaporin channels in the collecting duct. So that covers the really important parts of 11.3. Of course, there's more in the textbook that I didn't cover. But these were the most complex parts of kidney: osmoregulation and ultrafiltration. If you have any comments, please leave them down below, and see you in my next video.